Assalamualaikum warahmatullahi wabarakatuh. I, on behalf of my team from Biology Department of Andalus University and Ahmad Dahlan University, would like to present our research about effect of jicama fiber on energy intake and adiposity profiles in mice fed with high-fat diet. So, we are interested in investigating the medicinal effect of bengkuang or jicama. This tuberous plant is well known for having bioactive substances including flavonoid, terpenoids, and azoinulin, and also this plant contains high amount of dietary fibers. However, the study focused on the dietary fibers of this jicama remained less until recently. And in another side, that high-fat diet is a detrimental factors that could promote the development of obesity, diabetes, and associated metabolic disorders. So, in this present study, we attempt to investigate whether jicama fiber could be effective in precluding the dysregulated energy intake and adiposity caused by high-fat diet. So firstly, we isolate the fibers from the tuberous part of this jicama using the previous method as we published, and then we obtain this jicama fiber in powder forms. For the next steps, we test this effectivity of these fibers using the experimental animal models, biopsy mice, divided into 4 different groups based on the diet types. So the first group was fed with normal diet with very low fat concentrations, and then the other groups were fed with high-fat diet alone or high-fat diet supplemented with 10 or 25% of jicama fibers. Next, I will tell you about the result. So first, we found that a supplementation of jicama fibers could be effective in sustained normal body weight gains and also blood glucose levels in mice fed with high-fat diet. And the next, we investigated the epithelial white adipose tissue, and we revealed that supplementation of jicama fibers also could be effective in precluding the adipocyte hypertrophy and also reduce the fatty liver disease rates as shown by the gross morphology observations and also microscopic observations on the liver histology, showing that supplementation of jicama fibers about 10 and 25% could reduce the hepatocyte adiposity. Next, we also investigated the extrapancreatic adiposity and found that supplementation of jicama fiber 10 and 25% also could reduce it significantly and being comparable with those fed with normal diet. Next, we also measure the daily energy intakes and found that supplementation of jicama fiber could sustain the daily energy intake rhythms, and then also reduce the total energy intake in high-fat diet mice, and also sustain the energy intake ratio between the daytime and the nighttime. And also the most important also that supplementation of the 25% of jicama fiber could reduce significantly the energy intake during the daytime or resting periods. So in conclusion, our study revealed that supplementation of jicama fibers, but 25 or 10% could be effective in preventing the detrimental effect of high-fat diet, promoting the adiposity and also energy imbalance. And for the last, we would like to thank Indonesian Ministry of Research and Technology, the National Agency of Research and Innovation, for supporting us financially by multi-year basic research grants since last year until the next year. Assalamualaikum warahmatullahi wabarakatuh.